Welcome to this webinar on epigenetics. I'm Joel Exhibio. I am a PhD RD from Peru. I did my master's and my PhD in the US. I am a principal investigator for NIH. I have published more than 26 original research papers in the UK, in the US, in Brazil, in the Middle East, and in Peru. The first time I talked about epigenetics in the US was in 2007. I did a presentation on epigenetics and shamanism. People thought I was crazy because I didn't have enough evidence to support what I was talking about. Even though we have been using shamanism in Peru for more than 2,000 years to cure many diseases. Now we have a lot of evidence. So this is going to be just an introduction to the topic of epigenetics and shamanism. So let's start. What is epigenetics? So let's start talking about genetics. The Human Genome Project was released in 2001. We thought that we have discovered the cure for all diseases. Now we know that that's not true at all. The DNA is made of 4 letters. You get 3,000 million of these letters from your father and 3,000 million of these letters from your mother. It just takes one mistake in one of these letters to have a devastating disease. However, genetic diseases are very rare. We can count them with our fingers. Most diseases that we have nowadays are epigenetic, which means there is no change in the sequence of the DNA. The disease is caused by the expression of the gene. And it explains diseases, but also explains many other mechanisms. Like, for example, it explains your ability to build muscle. It explains your ability to deal with stress. It explains your ability to be successful in business, in social interactions. So it's not the DNA. You are not a victim. You are not a victim of your DNA at all. It's not in the sequence. It's epigenetic. So how it works, I'm going to give you a few examples so you can have a clear idea of what is epigenetics. So one of the examples of epigenetics is a worm. The worm eventually is going to transform into a butterfly. But there is no change in the DNA. Nobody is giving a new set of DNA, a new set of genes to the worm when it changes into a butterfly. So how is that two phenotypes, two bodies that look totally different, can have the same DNA? Well, that is explained by epigenetics. It's not changing the DNA. It's a change in the environment that produces the phenotype change. Another example of epigenetics are identical twins. They have the same DNA. However, when they start getting older, they start looking different. For example, I have two friends who are twins. They are now in their 50s. One of them lives in Peru. The other one lives in the US. The one who lives in Peru is normal weight. The one who lives in the US is obese. The one who lives in Peru is healthy. The one who lives in the US is a diabetic. The one who lives in Peru have kids who are also athletic. The one who lives in the US have kids who are also obese. So how is that two persons who have exactly the same DNA can develop different diseases and can pass these diseases to their kids? That is epigenetics. It's not the DNA, it's the environment what is producing a change in the expression of the DNA. All cells in the body have the same DNA. We have more than 200 different kinds of cells, and all of them have the same DNA. So a skin cell have the same DNA as a brain cell. However, they look totally different. A healthy cell have the same DNA as a cancer cell. However, they behave totally different. Is the environment what tells the stem cell to change into a skin cell or a brain cell? For example, if I have a stem cell and I put the stem cell in a petri dish, the stem cell will reproduce exponentially. But all the cells are going to be exactly the same. So if I start replicating these cells into different petri dishes, and I will change the environment, right? So I will change the environment. In a petri dish, I will put serotonin, right? A lot of nutrients. In another petri dish, I will put the stress hormones, like for example, cortisol. I will reduce the nutrients, right? Now the cells turn to be totally different, right? The cell in the good environment may change into a muscle cell, and the cell in the bad environment may change to a fat cell. So as you can see, both of them have the same DNA. What is producing the change is the environment. Now, in our bodies, what is the environment? The environment is the blood. So is the composition of your blood what produces the change? And how can we regulate the composition of the blood? Well, there are many mechanisms, but the most important ones are first, your emotions. Your emotions regulate your hormones. Your diet. Your diet changes the environment of your blood. Your environment, pollution, habits. So as you can see, diseases are not created by the DNA. We created a cancer. And if we create a cancer, we can also reverse the disease. If you understand this concept, you will realize that we can cure any disease if we can change the environment of the body. So the DNA is not a template. The DNA is an script that can be interpreted in many different ways. For example, here I have a soap opera. This is a Peruvian soap opera called Natasha. The original version was released in the 70s and it was a drama. The last version was released in the 90s and it was a comedy. So how it is possible that from the same script you can have two totally different productions? Well, it depends how the director is going to interpret the script. It's exactly the same with your DNA. Your DNA is an script. The director who is going to interpret the DNA is your brain, especially your subconscious mind. Your subconscious mind is going to produce emotions. Those emotions are going to release hormones. Those hormones will change the environment of your blood and it can produce a disease. If we go to the molecular level, this is how it looks. We have the DNA. The DNA is always around some proteins called histones. A proteins, right? You see here the A proteins. The DNA is always around these proteins called histones. Now, the histones have tails. You see these little tails? These are actually proteins, right? Glycoproteins that have the tails. Now, the gene could be expressed or inhibited depending on what molecule is attached to the tail. For example, alcohol in our blood. Normal level of alcohol in our blood is 0. We shouldn't have any alcohol. It's toxic, right? So normal level is 0. Let's say that you want to drink a glass of wine. Okay, now the yellow molecule attached to the tail, and that tells the gene that needs to produce the enzyme to detoxify the alcohol. So now the gene is on. Let's say that you keep drinking wine, right? Next day you have another glass of wine, but now you don't feel the same effect. So now you need to have 2 glasses of wine. Why 2 glasses? Because the gene that tells the DNA to produce the enzyme that detoxifies the alcohol is already on. So the enzyme is working, and now you need more alcohol to have any effects. And of course, because you are drinking more wine than yesterday, now more yellow molecules are attached to the tails, so more genes are activated. Now let's say that you stop drinking wine for several days. Now these yellow molecules are replaced by the purple molecules, and the purple molecules turn off the gene, so no enzyme is produced because there is no alcohol. Now we don't only have yellow molecules and purple molecules, we have more than 60 different colors, more than 60 different kinds of molecules that can activate or inhibit genes. And we have several combinations, so a gene could be partially activated or partially inhibited. Also, keep in mind that the DNA is the same for all the cells. So for example, your brain cell have exactly the same DNA as your red blood cells. However, the brain cell does not express hemoglobin, even though they have the genes for hemoglobin. Why is that the brain does not express hemoglobin? Well, because the genes that express hemoglobin are tightly packed. You see in this picture on the left, there are a lot of yellow molecules that are in a sequence. These molecules will tightly pack the DNA, so there is no expression at all. They totally suppress the expression of that gene, and that gene is going to be off forever. And that is why the brain does not express hemoglobin, even though the brain have the genes for hemoglobin. So that's how epigenetics work. It's not a change in the sequence. It's a change in the probability of expression of the gene due to the environment, like for example drinking wine, right? Due to emotions like happiness or fear. So that is epigenetics. Now we already said that the emotions play an important role on the expression of the genes. However, the emotions are regulated by the subconscious mind. We all know that if a kid have a stressful childhood, that kid usually is set up for failure in life. So when a kid uh, was raised in a dysfunctional family, for example, right, with a single mom, or with no parents at all. So this actually was confirmed in a study, in a beautiful study, using a model. The model are always mice, right? So the female mice, right, the female rat, shows love to their baby by licking and grooming. And some female rats are really good in licking and grooming, and others are not. So in this experiment, a mother that licks and grooms their baby was compared to a mother that is not good at licking and grooming the baby, right? So When the babies become adults, right? So when they are adults, stress was imposed. The stress was electric shock. So the cub that was raised by a mother that licks and grooms reacted in a positive way to the stress. They put electrical shock and the cub was totally relaxed. He continued living like nothing happened. He was the what we call the whatever rat. No stress at all. However, when the cub that was raised by a mother that didn't lick and groom was put under the electrical shock, the cub stressed out immediately, started shaking, started running, right? Was nervous. So you can see that the emotions and the reaction to stress is conditioned by the early years, by the early development, by the behavior of the mom. So the cub that didn't stress out released a lot of the happiness hormones, like serotonin, for example. And of course, is a rat that is going to be successful in life. On the contrary, the cub that was raised by a mother that didn't lick and groom have high levels of cortisol, stress hormones. And is going to develop a lot of diseases because it have a negative reaction to stress. So if we transfer this into humans, now we understand that a successful start in life is really important. And that's the reason why we always said in all our traditional culture in South America, we always claim that the key is childhood. If you take care, if you do a good job during the first 3, 4, or 5 years of life, your kids are going to be successful. You don't need to bother anymore. However, if you didn't take care of them during the first 4 or 5 years of life, they're going to be a problem forever. And this is just epigenetic. Now, many will claim that they have a really good childhood. They said, I was loved by my parents. I have a really good childhood, but still I have a lot of diseases. Still I have a negative reaction to stress. Well, it may be that the trauma is being carried on from generation to generation. For example, we all know that diabetes, type 2 diabetes runs in families, right? However, there is no gene that causes type 2 diabetes. So why is that my grandfather was a diabetic, my father is a diabetic, and I am a diabetic, if there is no gene that causes the disease? Well, what is telling me epigenetics is that there is a trauma that is being carried on from your grandfather to your father to you. And this was, 'This was 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 tested in a beautiful experiment again using mice. So the experiment was conducted in the US, and they have a male, right? A male rat. So the male rat was traumatized. They exposed the male rat to the smell of cherry blossoms, and after that they provide an electrical shock. They repeated this over and over again till the point that when the mice smell the cherries, the mice start shaking in fear because he already knows that electrical shock follows. So this traumatized male have an offspring. Now, the offspring was never exposed to the electrical shock, right? So the baby was never exposed to the electrical shock. However, when the baby smelled the cherries, the baby starts shaking in fear. So we can conclude that the trauma is being passed from the father to the baby, even though the baby was never exposed to the electrical shock. And that can explain why diseases are passed from generation to generation. Now the key is here. Can we stop this? Can we stop the trauma from being passed from generation to generation? And the answer is yes, of course we can, because it's not a DNA problem. It's not a genetic problem, it's an epigenetic problem, and epigenetic problems can be solved. However, in order to solve the disease, we need to fix the trauma. And this is the key. Diseases are caused by a traumatic event. What we are doing in medicine right now is just treating symptoms. And because we cannot fix the disease, we claim that the disease is a chronic disease that cannot be cured. That is false. We cannot cure the disease because we are not treating the cause. We are just treating the symptoms, the physical symptoms. The cause of the disease is emotional, is psychological. If we don't cure the trauma, we don't cure the disease. For those of you who want to read the original research, this is a reference. This is a beautiful study because they show what genes actually change in the brain in response to the smell of the cherry blossom, and also how the gene is passed, how the change in the gene is passed from generation to generation. So this is a beautiful study. You can read the study, you will understand more how epigenetics work. So the genes are expressed based on your environment, right? So nobody is born with a cancer, we create a cancer. Even when the disease runs in families, it's not a gene that is being passed on from generation to generation. It's the expression of the gene due to a behavior or to a trauma. For example, you may be a diabetic because you are addicted to sugar. You cannot stop eating sweets. But your father was also addicted to sugar and he passed the habit to you. It's not a gene, he passed a habit. And your grandfather was also addicted to sweets. And now you all three are addicted to sugar. It's not a gene, It's just a behavior. Now, it's a psychological problem because even though you know that sugar is killing you, you cannot stop using it. That's a very strong addiction, right? It's a psychological issue. It's not a physical problem. The physical part is just a reaction. So, how can we cure the trauma? We need to go back to your subconscious mind and fix the issue. Because your addiction to sugar was programmed during your first 5 years of life. Maybe you don't remember, but it is in your subconscious mind. So here is where the shamanism is related to epigenetics. In shamanism, we can cure any disease by going back into your subconscious mind and fixing the trauma. We use psychedelics. We call these sacred plants. We have been using them for more than 2,000 years. Psychedelics like ayahuasca, chacruna, washuma, San Pedro, tobacco, coca leaf. I'm going to give you an example. When I was a kid, I was walking on the fields and I saw a snake. The snake started moving towards me. And I was petrified. I couldn't move. So one of the workers killed the snake when the snake was actually very close to me. My dad said, we need to fix this. Because every time that you face a stressful situation, your body is going to remember, your subconscious mind is going to remember the snake, and you're going to be petrified. And this fear is going to make you develop diseases. So, I was maybe 8 years old, and he gave me washuma. And I saw a snake, a huge snake, because your fears are magnified under the effect of the psychedelics. So the snake was huge, and I was running away from the snake, and my father was telling me, don't run away, face the snake, face your fear. So eventually I faced the snake, and the snake swallowed me. I was inside the stomach of the snake, and my dad held my hand and said, I'm gonna give you a knife. Try to make a hole in the skin of the snake and get out. He didn't give me anything, he just held my hand, but he was talking to me, making me believe that I have a knife in my hand. So eventually I was able to make a hole and get out of the snake. So the image that I have in my head is this little kid on top of a huge snake, and the snake is dead. So I conquer my fear. Your subconscious mind cannot differentiate between what is real and what is fake. The last memory is the memory that is going to stick in your subconscious mind. And now the fear disappeared. And I was not scared anymore of snakes. And my body does not have any negative reaction when I feel fear. This is just an example of how we can cure basically any disease. Because most diseases are not genetic, they are epigenetic, produced by your emotions, which change your blood, which change your hormones, and eventually modify the expression of your DNA. So this video was just an introduction to the sacred plants, to shamanism. I'm going to follow up in other webinars when I want to go deeper into the techniques. and explain the science behind the techniques. So again, my name is Joel Exhibio. I am from Peru. I am from Trujillo, Peru. That's where I have my center. That's where I have my clinic. So stay tuned for the next videos. Have a wonderful weekend.